We're so glad to be able to offer this Research Highlights panel to celebrate the work of women scholars at the University of Virginia who are advancing the field of data science. My name is Claudia Schultz. I am the Associate Director for Research Development here at the School of Data Science, and I'll be moderating this panel. We have three fantastic UVA researchers joining us this morning. Each will give an overview of her work and share some recent results. We're very excited to welcome audience questions and make the panel as interactive as this medium allows. So please post your thoughts to Twitter using the hashtag 2020 Charlottesville WIDS, WIDS. Our fabulous events manager turned post multimedia producer, Danielle DeAndrea, will be monitoring the Twitter feed, so please ask plenty of questions. I'm pleased to present our first speaker, Dr. Cynthia Tong, Assistant Professor of Quantitative Psychology here at the University of Virginia. Cynthia received her PhD in Quantitative Psychology from the University of Notre Dame. She'll be sharing her work this morning on Bayesian growth curve modeling using conditional medians. Take it away, Cynthia.
Thanks, Claudia, and thanks to the School of Data Science for providing us such a great opportunity to share our research, especially in such a difficult time. I'm Cynthia Tong. Today I'm going to talk about my research on robust Bayesian growth curve modeling using conditional medians. First, let's talk about longitudinal studies. Longitudinal research provides innovative perspective to solve problems that are rooted over a short or long period of time. It helps us understand changes. Unlike one-off cross-sectional studies, which gave us information about subjects at one time, like a snapshot photo, longitudinal studies follow subjects across time, more like a photo album. They tell a story about subjects, not only at a moment in time, but also over time, showing how subjects have changed and what factors cause between subject differences in change. By using longitudinal designs, causal relationships can be well studied because the events happen first can explain the events happen later, but not the other way around. There are many longitudinal studies in reality. For example, if we measure participants' cognitive abilities across time, it's a longitudinal study. If we follow companies and see how their revenues changes over time and what factors influence their revenue, that's longitudinal study. And if we track countries or states and see how the development of coronavirus, and that's also longitudinal study. Here's an example of longitudinal study from the National Longitudinal Survey of Youth study, and school children's PIAT math scores were measured from the 7th grade to the 10th grade. This figure shows the trajectory plot for math abilities from 512 school children. Let's take a close look at this figure and by focusing on only 3 subjects. For the first student, we see that at the 7th grade and 8th grade, the math score is about 2.5. And the student worked harder at the 8th grade and get a better math grade. And then at the 10th grade, the score dropped back to 2 points again. For the second student, he got the score at 5 points at the 7th grade, and it dropped a lot at 8th grade. Maybe he's very unsatisfied with the grade, and so he worked much harder and then get a better achievement later. And for the third students, although we can see that the score dropped a little bit, but the students have a math ability almost always at the top. What I just talked about within subject change over time, for longitudinal study, we're also interested in between subject differences in change. From this plot, we can see that these 3 students have different initial levels and they have different change patterns too. The popularity of longitudinal research is growing in social and behavioral researches. Over the past few decades, there has been a considerable rise in attention paid to longitudinal methodology and application. A search of the PsycInfo database for longitudinal studies and panel studies gave us an estimate of the increasing popularity of longitudinal research. To analyze longitudinal data, growth curve modeling is very useful because it can directly investigate within subject change over time and between subject differences in within subject change. Although useful, longitudinal study faces methodological challenges, including handling data with outliers. To illustrate the influence of outliers in a data set, let's look at this simple example. We have data of salaries for 20 newly hired employees in a specific organization. From this table, we can see for each row, females have less pay than males. So we want to test whether females are underpaid by this organization. What we can do is to conduct an independent t-test, and the t-value is negative 1.889. So the p-value is larger than 0.05, which means it's not significant. We can say women are underpaid by this organization. However, later we figured out the last person in the males group, the salary for this person is 1,000 instead of 75. So given this new data, would you expect to see a more significant result or a less significant result? Intuitively, we would think the difference between the two groups are even larger, so we expect to see a more significant result. However, if we conduct the t-test again, and the p-value actually becomes 0.299, which is less significant. This is because 1,000 is an outlier in the data set. The t-statistic is based on the sample mean and sample variance, both of which are sensitive to outliers. And for this study, the outliers actually tend to increase the estimate of the variance and thus decrease the t-statistic and lower the chance to rejecting the null hypothesis. What I want to show here is that in this very simple example, only one outlier can affect our data analysis in an unexpected way. And in practice, we have more complex data and more complex models. For example, outliers may be multivariate and cannot be easily detected. And in this plot, the left panel shows longitudinal data without outliers, and the right panel shows the data with multivariate outliers marked in red. This is just the simulated data. By looking at these trajectory plots, we cannot easily detect the outliers by eyeballing these trajectories. If we fit a linear growth curve model to this data and with normal assumptions, we might have trouble seeing just as what we're seeing in this simple example. In fact, the rate of change was statistically significant originally, but becomes non-significant after the outliers were included. To reduce the influence of outliers, we propose to use robust method. We have developed a robust Bayesian growth curve modeling using conditional medians. It is well known that median is more robust against outliers than mean in measuring the central tendency. In the regression model, the regression line based on conditional means and conditional medians overlapped very well if there's no outliers in the data. But when outlier exists, the blue line, which is based on the conditional medians, stay the same, but the red line, which is based on conditional means, were actually affected by those outliers. Following the same idea, we propose to convert the longitudinal data analysis using conduct the longitudinal data analysis using the conditional medians instead of the conditional means. We skillfully use the asymmetric Laplace distribution to convert the problem of estimating a median growth curve model into a problem of obtaining the maximum likelihood estimation of this transformed model, so that the Bayesian method and the data augmentation techniques can be used for model estimation. Our simulation results showed that the new method performed similarly to the traditional method when data are normal, but outperformed the traditional method when there are potential outliers. We conducted a real data analysis. We analyzed the data from the Virginia Cognitive Aging Project, the VCAP project started from 2001 and is currently one of the largest active longitudinal studies of aging involving comprehensive cognitive assessment in adults in the world. So in this study, we focus on the development of vocabulary abilities, and we use a subset of the data. As you can see from this table, the traditional mean growth curve model and the median growth curve model gives us different results. And we trust the results from the median growth curve model because there are potential outliers. And the results is more reasonable and interpretable as most participants in the data set accurately. Remember at the beginning, I said there are many methodological challenges for longitudinal research. Although we focus on the normality issue in this study, but the proposed method can be extended to address other issues and can be applied to other models. I welcome all the questions and comments regarding this study, and I'm ready for the question.
Hi, everyone. Are we ready for questions? Okay. I have a question, Cynthia. Mm-hmm. You mentioned that you've applied this approach to the Virginia cognitive aging study.
Yes.
Could you talk a little bit about that data set and about how should I put this? About the questions from cognitive psychology domain that can be answered now that you've applied this growth curve modeling to the problem. So what do we know now that we didn't know before because you applied this technique? Sorry.
Mm-hmm. So the new method is like have the techniques to, have the ability to answer all the questions that we were able to answer before. But the thing is, like, this method is more robust and more stable. Even when your data is not normally distributed or you have some noise in your data, the method still works very well. Mm-hmm. As you see, I showed like one outlier can affect the data analysis in an unexpected way. Mm-hmm. And this method definitely gave you more reliable results. Mm-hmm. But also this method have some other abilities in explaining the results. For example, this method can be extended to do the quantile growth curve modeling. In that way, you can study people's behavior or people's ability at different quantiles instead of just look at the overall change patterns.
Mm-hmm. So what are some of the domains where this problem of outliers in longitudinal data are most pronounced in your experience?
I think with longitudinal data in social sciences or behavior sciences, it's just these kind of data are very messy and everywhere. Like, we need to use robust method instead of just simply use the traditional method. At least we have to do some sensitivity analysis to see whether the traditional method worked well. So as I can see, like in psychology or marketing and many other studies, the data set are really not normally distributed. In that case, we may at least have to do different analysis and see if the results are the same, or just simply use the robust method I developed.
Great. Mm-hmm. You mentioned the application right now in marketing. Mm-hmm. You mentioned the National Longitudinal Study of Youth, so education, mm-hmm. The cognitive aging, so cognitive psychology. And you also mentioned at the beginning the application to our understanding of the current pandemic. Mm-hmm. Could you speculate a little bit about some of the issues facing data scientists in the context of this pandemic with regard to quality of data related to COVID-19?
Just about the COVID-19. Yeah, I'm actually very interested in that data set. And yesterday, I just submitted a request form to get the data set from a website which have all the data sets about the COVID-19. And to me, I feel like it's very interesting to study how does different states or different countries may have different change patterns and how the confirmed cases grows across time. And of course, that's definitely the data not normally distributed. I'm interested in see if there are different patterns of growth and whether they are latent classes or latent groups. Like some countries might share some pattern, but some other countries might share a different pattern. I'm quite interested in applying this method to analyze the data.
Yeah, so you have an emerging research agenda related to the pandemic?
Yeah, at least I'm quite interested in that. So I requested to on the website and to get the data set. With the data set in hand, we probably will do some analysis.
Okay, great. And some interesting opportunities for interdisciplinary collaboration, it sounds like, too.
Cool, yeah.
Yeah. Well, thank you so much for sharing your research with us this morning. We're going to go to our next panelist, Dr. Sana Sayed, who is Assistant Professor of Pediatrics here at the University of Virginia. Dr. Sayed is a global health scientist with a background in pediatrics, gastroenterology, and nutrition, and epidemiology. We're very proud to say she's also a student in our Masters of Science and Data Science program. And if you were watching earlier, you'll recognize her from the video about UVA's collaboration with the Met Museum, which we aired just before this panel. She'll be sharing her work this morning on computational image analysis. The floor is yours, Sana. You'll need to unmute yourself.
Yep.
Great.
Hello, everyone. Thank you for this amazing opportunity, and kudos to the organizers for pulling this off. So I'm just going to start sharing my slides. Okay. Can everyone see them? Claudia, can you see them?
I can see them. Great.
Okay. Excellent. So as Claudia mentioned, thank you for that introduction. My name is Sana Sayed. I'm a pediatric gastroenterologist here at UVA and a current student. The title of my talk is Machine Learning for Image Analysis of Inflammatory Enteropathies. So before I go into my talk, this is what I deal with, the human gut. So when I talk about enteropathies, I wanted to take some time and explain what part of the gut I'm talking about, which is really the small bowel. So the small bowel is this long tube, and it's covered with these finger-like projections that you see as villi over here that help increase the bowel's capacity to absorb nutrients, which is really what we need to grow. So there's this progression that happens from these healthy intestines to enteropathies, and there'll be two that I'll be talking about. One is celiac disease, and the other one is environmental enteropathy, which is a global health problem. So the context of this whole work that we're doing is sort of summarized in this slide. So as clinicians, when I see patients in clinic or in the ER or elsewhere, we gather a lot of data on our patients. So we have growth measurements, we take blood, urine, stool samples. And so all this clinical data helps us make a decision whether we need further, more invasive testing. So these biopsies that we get, which is the picture on top, is pieces of tissue that you get from doing an endoscopy, which is a tube that you put down a person's mouth to take pieces of tissue. And so most clinical diagnoses at this point are based off this pink tissue, but we now have the ability to query this tissue further using molecular methods, which is this panel on the other side. So multi-omic data means the ability to look at RNA expression in tissue, which is transcriptomics, look at specific metabolites, which is metabolomic analysis, and to look at microbial analysis of what's in your gut, which is the microbiome. And the overall goal of all of this work is to see if we can find disease patterns which link with specific phenotypes of patients for any given disease. So, and then the goal is to give a treatment and management plan that is individualized for each patient. So that was a lot of words, and I thought it would be nice to put it together in a story. So Matt is a 2-year-old. That's not really his name. That's not really his picture. 2-year-old who came into my clinic about a year ago. So he's 2 years old. He comes because he's having difficulty growing. He's having diarrhea. He has this big distended belly, as you can see. And so from my perspective, what I was most concerned about, and just as an FYI, his weight was roughly what my 1-year-old weighed. So he was very behind in terms of growth. So the biggest thing I was concerned about celiac disease. So we did that, did the relevant testing, and we diagnosed him. So what is celiac disease? Celiac is an enteropathy. It affects about 1% of children in the US, and it's permanent gluten intolerance. So gluten is something that's present in wheat along with a couple of other items, and it causes intestinal damage. And children like Matt suffer from diarrhea, abdominal pain, anemia, decreased bone density, and has an overall increased risk of intestinal cancer. So I was telling mom all of this, The risk of intestinal cancer is if you don't remove gluten from your diet. So I'm telling mom all of this, and I say, well, and up to 10 to 30% of children with celiac disease will go on to develop autoimmune diabetes or thyroid. And she said to me, well, what does that mean for Matt? Will he 100% get it? Is it a 100% risk for him? Is it a 1% risk? And so that's really what I mean about developing an individualized profile for a child. And that's not where we are right now in GI disease. Moving on to the other enteropathy I'll be talking about. So this is a global health problem. The picture which shows these children, this horizontal line, that tells you an average height for a healthy 5-year-old. And you can see how below that line these 3 children are. And this is really a condition of poverty. It occurs when children are exposed to repeated episodes of diarrhea due to unclean water and poor hygiene. So this is where's Waldo, right? And this is what human pathologists have to deal with. So current GI diagnoses is about looking for rare events in a sea of tissue. So our question was, can machine learning help characterize key disease features in celiac disease and environmental enteropathy with the goal of sort of that precision medicine, individualized care model? And from a clinical perspective, we wanted to see if we could break apart and understand the decision-making of that machine learning architecture, which is convolutional neural networks. So in our initial model for celiac disease and environmental enteropathy, this was the broad approach we followed. So we had highly digitized images. So the size of a biopsy that for a GI biopsy is typically 1 to 2 centimeters. But when you digitize them at high resolutions, we have about 20,000 pixels by 20,000 pixels, which means we need to batch them into these tiles, which were about 500 by 500. And then these get fed into a convolutional neural net, and this is the one we used, which was a ResNet 50, and paired with an outcome of what is the probability of celiac versus environmental enteropathy, EE versus normal. The other thing, like I said, we wanted to see how the model was deciding its final probabilities. So this is an example of a dog and a cat type decision to figure out a striped cat. This method called GradCams, developed by a group in Georgia Tech, looks at the stripes, and here it looks at the dog's face. So when we looked at, so overall networks accuracy in classification was about 92, 93%. And so for celiac disease, it was looking at crypt cross sections. So at the bottom of the villi, there's a part which generates and gives rise to healthy tissue. And so in disease profiles, there's crypt hypertrophy. So very appropriately, it was looking over there. There's a lot of inflammation, so it was looking over there. In environmental enteropathy, it was looking at slightly different areas. So it's looking at inflammatory cells in the sort of the top part of the villi. And then it has these focus on the top part, and so it was ignoring the crypts at the bottom. And most interestingly, in the controls or normal healthy patients who were scoped for other reasons but had normal tissue from inside, it was looking at sort of the edges and what looked to be otherwise normal tissue. So how does that all feed into that question of Matt? Well, after we ran our initial models, we then did a celiac specific model. And here what we did is we looked at disease severity in celiac. So we went from mild to most severe celiac. And we tried to see if the ML model could help pick apart cellular level differences in the tissue. And what we found was in children with the most severe type of celiac, it was picking up these cells that are actually not part of typical celiac diagnoses. And when we went back and did a chart review, those children had either autoimmune diabetes or thyroid disease. So that in a nutshell is the kind of insights we hope to find. Now, that celiac enteroendocrine diabetes signal, that's the prelim data. We're confirming that from this multi-omic pathway. So we're doing immunohistochemistry and RNA-seq. But that's really what the goal is. So you see disease patterns linking with Matt's specific type of celiac, so that we're able to tell him what is the likelihood or risk for him to get diabetes and his personalized management. So as we know, any kind of very ambitious big project involves a large team. So this is our team. I co-lead the Gut Intelligence Lab with Dawn Brown here at UVA. And we have systems engineering, PhD students. We have MSDS students and now data scientists. We have medical school graduates, UVA medical students, and UVA undergrads in biology, along with key mentors and collaborators in gastroenterology, pathology, and global health. So I just would like to end with a slide to say, I know these are uncertain times, unprecedented times, and I really do believe in the power of of us all to solve this problem. And I think data science will be one of those key tools that we use to do so. So on that note, I would like to thank you all and ask if there's any questions.
Thanks, Sana. Could you talk about, you probably know I'll ask this, about what you learned in your capstone project with the Met Museum and how that's influenced your work in medicine, either in relation to this project or others?
Yeah, so the Met Museum project was very interesting. I picked it because it was image analysis. So the kind of approaches to using a ResNet 50 to look at human tissue, we used similar approaches in studying the Met Museum's data. Now, my purpose in doing that capstone was to understand image analysis better. But what I learned in the Met project was things like, you really don't know what the project leads to you towards. So for example, in the Met project, we ended up exploring bias in the data set. And we looked at how the CNN was classifying men versus women. And then we used GradCamps to explore the misclassifications and the classifications to see what the network was looking at. And these were pre-labeled data sets by humans. And what was really interesting was things that we are aware of, right? So it uses not just the women's long hair to diagnose, but often what they're doing or the posture in which they're sitting or what they're wearing. So then a lot of men who are wearing long clothes or long dresses, such as in South Asia, were getting misclassified as women, or Native Americans with long hair. So it's a little bit of cross-pollination from both worlds. So I've been sensitized more to the idea of bias in our medical data sets.
Interesting. With a lot of image analysis, a key component is the training and labeling.
Mm-hmm.
Can you talk a little bit about how that was accomplished with these
Medical images?
Gastro image data, yeah.
Yeah, that's actually a Herculean task, really. So it was done in sort of two levels. One, we used a medical chart review to decide what the overall diagnosis was. So whether a patient had celiac disease, whether the tissue was called normal. Now, there's a subtlety to the celiac diagnosis in that you sort of take biopsies from different spots in the bowel when you're doing a scope. So even though the patient has a diagnosis of celiac disease, some of the biopsies could have been normal, which was very confusing to the CNN model when we initially started. So the second level of labeling was we went back and reviewed each and every single biopsy on every patient that we had. So then did sort of class level labeling. So overall diagnosis was this, but the individual biopsies were this. Mm-hmm. And then once we have GradCamps to interpret, so my research assistant, who's a medical graduate, she and then I review them, and then we review them again with the clinical pathologist, or in collaborator here at UVA is Chris Muscalic, who's the chair of pathology. So there are many levels of review and labeling.
Okay. Could you give us a sense? I'm always curious about the human labor embodied in these models, right?
Yeah.
You know, how many files, how many person hours, what level of expertise is required to make those classifications or labels?
Yeah. So for example, Chris Muscalic or myself, to be like a practicing physician in the U.S., you need to have gone through undergraduate medical school, typically 3 to 5 years of a residency, and then 2 to 4 years of fellowship. So that's all the years it takes you to be able to be the final expert who reads. So that's just in terms of that it's hard to find people to do that. The other thing is for humans, right? So you can just sort of scan through a biopsy and say, overall, this is celiac and be done with it. But when the CNN gave us GradCamps, it gives us, for each run of the model, you can pull out as many as you want. So at one point we wanted to group. So it's sort of like it's an unsupervised method in that it gives you GradCamps and then you have to group them to say, well, this is the biological meaning of this. This is a biological meaning of this. So you have to really review a large number. So we reviewed anywhere between 500 to 800 GradCamps. This is on top of all the other labeling that had been done. So, I would say it would take my research assistant 10, 20 hours. Then it would take another, 2 to 3, 4 hours for me to review with her. And then another couple hours for the pathologist to review with us. So It's hard because at this point, I don't think, so humans are capable of doing diagnoses. There's no pathologist who can't do celiac or normal or environmental enteropathy diagnosis. The problem is that to look for those subtle hints. So if you ask a human to say, can you look at across all celiac biopsies that you've looked across, and can you find any subtle cellular signatures? So any specific cells that are missing in or present in patient with diabetes or patient with refractory celiac. That would be hard for a human to do, but very easy for a CNN model to do. But to create that CNN model is all this upfront work.
Okay, that's fascinating. Thank you so much for sharing your work with us, Sana.
You're welcome, and thank you for the invitation.
Now we'll move on to our third and final speaker, Dr. Behnaz Moradi. Behnaz is a postdoctoral research associate here in the School of Data Science, working with iThrive, a clinical and translational science program that brings together research and clinical data from across the Commonwealth of Virginia to improve health outcomes for all. Behnaz will be sharing her work on using cyclic structure to improve community detection and statistical inference in network data. Please join me in welcoming Behnaz. You might be muted.
Okay. So thank you, Claudia. And I'm going to introduce my novel method for incorporating cyclic structure of the graph to improve community detection as well as statistical inference on network data. So I'll begin with defining communities or cluster, which are any subset of the closely connected data objects. And the individuals, they're connected in a way that individuals within the same communities have similar characteristics and behaviors. And variability across communities or clusters is significant than between them among clusters. So researchers typically want to know who belongs to which communities, or like a bigger question, if we do have a community structure at all. So since detecting communities and clustering is an NP-hard problem, therefore we're limited to using heuristics to do this. Two popular heuristics that has been used broadly for large networks are CNN and Louvain method. Most of these methods work with modularity maximization, and they only capture the graph modularity. So it has been observed that these methods can be improved by incorporating additional graph structure through the edge weights. So some of these graph structures could be running a random walk or counting number of triangles or cycles. But we're particularly interested in the edge weights derived from the cyclic structure of the graph. And the idea is that we think that if edges are more prevalent within community than across, so cycles could be more prevalent within community than across. So if you imagine two groups with the same number of the edges, we think that the group that has more cycles has more community structure than the other. And this is actually coming from the idea of information circulation, which we prefer the groups with more number of the cycles. So from here, I'm going to introduce my method for community detection, and namely renewal non-backtrack random walk, or what I call run broad, throughout this presentation. I will begin with defining non-backtrack random walk, which is an important variant of random walk in which the walk is prohibited from returning back to the previous step in just 2 steps. This graph shows a non-backtrack random walk, which initiates the walk from the starting node, and then it cannot backtrack in just 2 steps, but it is free to backtrack in other steps. So what is RNBRW? RNBRW is a non-backtrack random walk that terminates and restarts once it visits a retracing edge. The retracing edge is the dashed edge that you see in the graph, which is the edge that completes this cycle. We think that retracing edges are critical in the formation of the cycles. So we use RNBRW for community detection as well as hypothesis testing on network. We use RNBRW for community detection by weighing the modularity function by retracing probability through CNN and Louvain. And since obtaining these retracing probabilities is hard analytically for general networks, we use Monte Carlo simulation to estimate the retracing probability more efficiently. And the probability of an edge being a retracing edge in a renewal non-backtrack random walk is approximately proportional to number of the times each edge has been retraced by a non-backtrack random walk. So a very nice property of our algorithm is that each run of our algorithm is independent of the other. So we can collect samples in parallel, and this leads to a fast convergence. We further establish the limiting distribution of the largest eigenvalue of properly shifted and scaled adjacency matrix weighted by RNBRW. And we use it for, we use this distribution for our hypothesis testing to detect the community structure rather than the communities. So common approaches to do the hypothesis testing looks at the maximum eigenvalue of the adjacency matrix. And the larger this value, the better community structure. And we construct our hypothesis by considering a homogeneous graph such as Erdős-Rényi, which doesn't have any community structure as our H-null, and stochastic block model or any other type of block models for the alternative hypothesis. So we use edge universality, which states that the distribution of the shifted and scaled large singular value of a Wigner matrix, here it's proved that adjacency matrix of an Erdős-Rényi is a Wigner matrix, converges weakly to a Tracy Widom distribution with parameter beta equal to 1. And after showing that adjacency matrix weighted by RNBRW is a Wigner matrix, we use the result of edge universality, and we try to construct our hypothesis. By standardizing our hypothesis and analytically obtaining the mu or the expected value for the RNBRW edge weights, and then estimating the sigma using a bootstrap sampling, we then shift and scale the largest eigenvalue of this Y star, and then our result shows that theta star basically converges weakly to a Tracy Widom distribution, but it's not a great approximation for the small sample sizes. Therefore, we use bootstrap Bartlett type bootstrap correction on the theta star to match the first and the second moment of the Tracy Widom distribution. And then we compute the p-value using the Tracy Widom quantiles. So this graph shows the comparison of RNBRW, Louvain weighted by RNBRW, and it shows that it outperforms other scalable community detection algorithms substantially, as well as unweighted Louvain. This graph also shows the challenging the challenge of sparsity in the network and community detection. So typically, the existing community detection method suffer from the sparsity. And so therefore, I challenge my algorithm with different average degrees of in order of log n, 2 log n, and 3 log n. And my simulation results shows that Louvain and CNN weighted by RNBRW not only outperforms unweighted Louvain across different average degrees, but also it outperforms across different network size as well. So this graph also shows the comparison of different weighing method with RNBRW. So my simulation results shows that the CNM weighted by RNBRW outperforms CNM weighted by other weighing methods such as KDV, K-path algorithm, and SIMRank. These algorithms are based on random walk and other graph structures. And my algorithm is actually robust across different network sizes and average degrees. In this table, we also challenge our method across a different average degree and different network size for different community detection algorithm. And our algorithm actually outperforms all this algorithm in most cases, and we have comparable results for some of them. So in this graph, as we see, the right-hand side graph shows the benchmark weighted by RNBRW, and we haven't done any community detection process here. It's only the edge weights by RNBRW, which as we see, the right-hand side graph has much more distinguishable community structure than the left-hand side. So this graph only also shows the comparison between Tracy-Widom distribution, the true Tracy-Widom distribution, and the distribution of our test statistic under the null hypothesis or heterogeneous graph model. And as we see, we have our curve are kind of close to each other, but it's not a perfect approximation yet, and we're still working on this. Thank you so much. And I am ready if there is any questions.
Right. Behnaz, if you could stop sharing your screen, that will give control back to the producers.
Okay.
Great. I wonder if you could talk a little bit about, or speculate, I suppose, about how this technique of community detection could be useful in understanding the epidemiological data that's being generated in the current pandemic.
Yes. So in the ongoing, pandemic, we are facing a much more dispersed population, where the population is not densely connected anymore, and it rather has a more community or modular structure. Mm-hmm. So the effective community detection and at the level and making the policy at the level of the communities can actually reduce the spread of the disease, in my opinion. And also, as far as the cyclic structure and the COVID cyclic structure in the contact network, I think this cyclic structure in the contact network will increase the risk of the infection. And by detecting, if we have given the contact network, finding the cycles could help us, to find, to decrease the risk of the infection and spread in the network.
We've seen some countries doing a better job than we do in the US on contact tracing. For example, in South Korea, they've been able to identify those clusters to really significant effect. I wonder if you could speak to the utility of that, both from the data science perspective and from the medical perspective, if you care to.
Yes. As far as the obtaining the contact network, unfortunately, as you said, we don't have it here, but they do have it in South Korea. I haven't been able, to access that data yet. But having those data are really, really helpful as far as the modeling, modeling the pandemic, and as well as, knowing and gaining more knowledge about the structure of the network and, making the policy accordingly. For example, if we could know the clusters, say, and in this cluster, the spread, is much more intense than the other clusters, maybe quarantining only this cluster, and having a more, restrict policy and quarantine policy in these clusters, would be more beneficial. So I think, having that knowledge is substantial as far as, the modeling and to help interrupting the epidemic.
Okay. Well, thank you so much for sharing your work with us. And thank you to everyone in our audience for joining us. Please join me in thanking our fabulous speakers. And also join us in thanking the production crew from the AV company, Arlen Burgess, Danielle DeAndrea and her team, Cody, Meg, Connor, and others who've been helping to support the presenters behind the scenes. I also wanted to thank everyone who joined us for this session. We had over 600 people register for WIDS this year, which is more than twice the number we could have accommodated in the physical space that we had originally reserved for this event. In our current crisis, the work of data science researchers is vitally important. We've seen the role that data has played in informing policy decisions and advancing our understanding of this disease. The School of Data Science is in the process of pivoting our research efforts to support COVID-19 response. So please continue to monitor our website and social media feeds in the coming weeks for updated information. This concludes the Research Highlights panel. We will transition now to some pre-recorded content for you to enjoy during lunch. Please make sure to join us this afternoon as live discussions will resume at 1 p.m. Eastern Time. Continue to monitor the hashtag 2020 Charlottesville WIDS for the latest updates and links. I'm wishing all of you health and safety from all of us here at the School of Data Science. Be well.